In this video I'm going to cover the anatomy of the retrosplenial region as shown by Dr. Brodmann. So Dr. Brodmann, you can see his photograph right there, was a German neurologist who revolutionized the study of the brain by creating a brain map which was grounded in the microscopic properties of the brain. For the first time, researchers and clinicians alike had access to a detailed map of the brain, which was described in relation to the large brain structures such as the grooves, that's the sulci, and the ridges, that's the gyri. Brodmann had assembled a significant collection of anatomical specimens from many species. This enabled him to compare the brain maps of different species, and this in turn helped him to better understand a human brain map. Brodmann's description of the retrosplenial region is a case in point. Brodmann writes that the microscopic anatomy of the discrete areas he found were confirmed by him only after a comparison with the brain maps of other species. So what he was saying really was that it was really difficult for him to identify Brodmann areas 26, 29 and 30 were it not for the fact that they were more well developed in other species, and he knew that they were there in these other species, and this gave him the clue that he needed to describe them in humans. An important feature of the brain maps are the eponymously named Brodmann areas. These are the discrete areas within the brain regions that Brodmann described. These areas are distinguishable by the structures formed by the neurons when viewed under the microscope. They give characteristic patterns of the layers of the cells, and this was the cornerstone of Brodmann's analysis. Brodmann's analysis of the human brain is confined to 21 pages in one English translation. That's just 21 pages. Now, what's interesting about Brodmann's analysis is that he doesn't include many photomicrographs to illustrate the Brodmann areas. Indeed, for a work which has had so profound an impact, the relative brevity of the description is remarkable. Nevertheless, it's provided a robust model of the brain for researchers and clinicians alike for the last 100 years. So turning to the retrosplenial region, Brodmann's description contains 3 Brodmann areas. Brodmann area 26, the ectosplenial area, Brodmann area 29, the granular retrolimbic area, and Brodmann area 30, the agranular retrolimbic area. Now I'm going to scroll down. I'm going to show you some pictures as I'm discussing this so you can get a better handle on where these places are with reference to the surface anatomy. So this is a replica of one of Brodmann's original images in his 1909 work. This lower diagram is the medial aspect of the brain, and we can see 26, 29 and 30 there. Now what's really interesting is, this diagram is quite popular among people studying neuroanatomy because it's a kind of iconic brain map or brain diagram. But actually in his book, Brodmann writes that his illustration of these 3 areas in this diagram is actually exaggerated. He made them a lot wider than they were in reality, and that's because he wanted the reader to better understand the relationships. So it's a bit like the London Underground map. It's not a real representation of actual distances, it's an abstract representation, at least in this part. So due caution should be taken when looking at this brain map and interpreting the Brodmann areas. And just to point out the earlier discussion about the structure of the cells and the layers, this is a famous neuroanatomist that really started the microscopic study of neuroanatomy, and his name is Professor Ramon Cajal, and here you can see the different layers when viewed under a microscope. These are drawings of the microscopic assessments. And these are different cells, different cell bodies that are stained in different ways in the different diagrams. Anyhow, moving on. When we talk about the retrosplenial area, one of the important things is the relationship of these areas to a structure in the brain called the corpus callosum. And the corpus callosum is an incredibly interesting structure which basically connects the two sides of the brain. And there's some people who have had colostomies. They've had the corpus callosum removed, for instance in the treatment of epilepsy, and there have been all sorts of studies on people that have undergone these operations, telling us a lot about what the corpus callosum actually does. Anyway, here's the video I've created from public Commons, Creative Commons video. Okay, moving on to another diagram. This time, this is a replication from a 1918 edition of Gray's Anatomy. Here again, we see 26, 29 and 30 towards the back of the corpus callosum. And what happens is that Brodmann describes these as semicircular areas, each encapsulating the previous one. And also he describes these three areas as corresponding to the isthmus of the cingulate cortex, which is illustrated in this diagram, which is from a 2008 PLOS biology paper by Hagman and colleagues. You can get the link on the blog. Now there's a previous post, and in fact a previous video on the cingulate region, in which I've discussed another part of the cingulate cortex and just pointed out some aspects of Brodmann's description of these other areas in relation to the precuneus. Anyhow, we'll cut to the chase, and we're here now, area 26, the ectosplenial area. Now, under the microscope, Brodmann said that this was a really poorly developed area. He didn't have the kind of refined structure that was visible in other parts of the human brain, and indeed in other species. So in other words, in other species they had a more organised structure than in humans, which I find quite curious. Another point about the ectosplenial area is that it lies within a sulcus, a groove, the callosal sulcus, related obviously to the corpus callosum. And that BA26 is related to the posterior, the back of the corpus callosum, and forms a semicircle. The next area is area 29, which is the granular retrolimbic area. And he said this has got a well-developed inner granular layer, but a poorly developed layer 3. So what he's saying is if you look under the microscope, you can kind of recognise this area because one little bit is poorly developed and the other bit is well developed, and that's why he gave these areas distinct labels. And the granular retrolimbic area surrounds area 26. Next up is area 30, which is the agranular retrolimbic area, and Brodmann writes that this time layers 3 and 5 are well developed, but layer 4 is poorly developed. So again, these are very subtle distinctions between these three areas. You have to look under the microscope, and when you do, you have to look at certain layers of the cortex in these areas in order to distinguish them. And he could only be very confident about these distinctions when he looked at other species. So there's a lot of triangulation that's going on here. And what he says is this is the edge of the isthmus. So this is the isthmus in this diagram. He said that Brodmann area 30 not only surrounds these two areas, but is also the edge of the isthmus of the cingulate cortex, and that it extends onto the anterior aspect of the calcarine sulcus. So that's it. That's all for these three Brodmann areas, which form the retrosplenial region.